All right, we'll get started on the blood system. We'll most likely finish up next time we meet. All right, 4 to 6 liters. That's about a gallon and a half in most people. So, of course, carry oxygen, O2, and nutrients. Removes waste, CO2. Hormones. So, the last week we'll talk about hormones. 2 types of hormones. And then vasodilation, constriction, we alluded to in the cardiovascular chapter for temperature regulation. What is the pH of blood? What is pH? pH is a scale from 0 to 14. So 7 is neutral. All right. What is acid? Low pH or high pH? Acid is low pH number. Base is a high pH number. So what's the pH of blood? 7.4. If it's outside 7.4, we die. Right? So we have to maintain our blood pH at 7.4. Things we put in must keep it at 7.4. If we take stuff out, we better keep it 7.4. Urethro. What color is urethro? Red. Red blood cell, you notice how they're kind of concave? That's because there's no nucleus, no DNA. So really they're just like simple machines circulating throughout the body. That's all they are. And once they get damaged, broken, and used up, they get filtered out and replaced. Hemato is blood. Crit means to separate. So when you separate the liquid and the solids, you get about 45% of the solids. In between the liquid and the cells, you get this white coat. And they just named it by what it looks like. And a lot of the white blood cells are there as well. You'll do your peripheral blood smear in one of your lab classes. You'll get a drop of blood, you'll feather it out, you'll stain it, you'll identify the different types of blood cells. White cells, about 1%. What's another name for white blood cells? Luke. Luke is white. Just think of Darth Vader. Luke, I am your father. The liquid part, 55%. Right? So the hematocrit is what's separated from the blood, the solids. So you can tell if somebody's dehydrated or if they're making too many blood cells based on this ratio. Hemo, blood again. Goblin, that's just a protein. In this case, it's a globular shaped protein, and what this guy can do is it can carry oxygen. So on hemoglobin, you have a little iron. So you guys heard of anemia? If you're anemic, they like to give you a lot of iron. So a lot of times you have an iron deficiency as well. If it's carbon dioxide, then it's deoxyhemoglobin. What color do you think oxyhemoglobin is? Reddish, right? And then deoxyhemoglobin is probably that bluish color. RBC, red blood cell. So within 1 ml, tons. This is the most common blood cell type. And when you do your differentiation, that's all you see, mostly red blood cells. A is without. Anemia is a blood condition. So this is any blood condition where you're without cells. In this case, it's without red blood cells. We have 2 types of bone marrow. We have red and yellow. Yellow is where fat is produced and stored. Red is where our blood cells are produced. And all of our blood cells come from a stem cell like, and they just change and they make all of our different cells and platelets. So it's pretty cool. If you donate stem cells, they don't have to get it from your bone marrow. They can just spin it out from your blood, and then you can donate that instead of getting an invasive procedure, harvesting it from the bone marrow. They don't live too long because there's no DNA, so there's no repair mechanism. So they get damaged, bruised, bumped, they just get filtered out. Urethra is what color?
Red.
This is the hormone, poietin, that stimulates the production of red blood cell. EPO, or epogen, is the brand name. If you have kidney failure, guess what you're going to have also? Anemia, because your kidney produces the hormone that stimulates red blood cell production. Right? So red blood cells help you carry oxygen, increases your stamina. You guys ever heard of Lance Armstrong?
Mm-hmm.
So, who is Lance Armstrong?
The guy who got caught for dosing.
No, he never got caught. No, he never ever got caught on any testing, any screening. He passed them all because those screenings are not a drug test. They're an intelligence test. If you know how to use steroids, human growth hormone, epogen, and stem cells, and whatever else concoction they were using, they all cheated. Everyone had to cheat. So it was a dirty sport. If you gave it to the next clean guy in the Tour de France, you had to go down to like 30th place to find somebody who was clean in that sport. But this is blood doping, one type of blood doping. So instead of training at Big Bear, right, this guy cheated. He just injected himself with this hormone, produced more red blood cells, carry more oxygen, more stamina, 7 Tour de France's in a row. As mentioned, we need iron, so they'll give you an iron supplement. So that iron in the center of hemoglobin helps hold on to oxygen. B12 helps you produce good red blood cells. So if you're not absorbing B12 or your gut bacteria is not producing it for you to absorb, then you might have anemia as well. Folic acid is another B vitamin, and most of the B vitamin are involved in your body's energy processing and management. So anybody had a Monster energy drink lately? If you read the back of that, you'll just hear nothing but B vitamin, B6, B12, B4, B6, riboflavin, thiamine. And then when you urinate, what color is your urine after a Monster? Bright yellow, right? Neon yellow, because that's all the B vitamins going through your body, because B vitamins are water soluble. They just pass right through you. Who feels hot after a Monster energy drink? One B vitamin is actually niacin, and they use that to reduce blood pressure because it opens your blood vessel. So you feel a little flush after taking niacin. What's that?
Isn't that why when you take too much niacin and it comes out in your pores, it kind of burns a little bit?
I'm not sure, but yeah. You experienced the burn from niacin?
No, we had a couple guys in the Corps that overdid it on niacin, and they sweat, and they kept saying that they kept burning.
Okay, yeah.
Wow.
Yeah, because that's sending the heat to your skin, the top of your skin, because that's what it's designed to do. Because they give it to people with high blood pressure, and then it mimics like having a hot flash. Exactly. If you want to know what getting a hot flash is like, just pop some niacin.
They're not fun.
Not fun. White blood cells, lot less. So most of the cells you'll see, like all these cells, those are all red blood cells. A majority will be red blood cells. You're going to have to scan the whole slide for white blood cells. So here's an example. We have granulocytes and agranulocytes. You know it's a granulocyte because they're filled with granules, right? They have these little dots. So neutrophils, they kill bacteria. We have eosinophil. What color is eosino? What color is that? It's another term for red. And then lastly, basophil. Right? This is part of your allergy response, basophil. Eosinophils, parasites. So these guys will be filled with dots. This guy is neutral when we stained him, so he didn't color anything. This guy stained red when we colored him, so we called him eosinophil. And then this guy was basophil, so it colored if we use a basic environment. So they just named it by how they got them to show up. So those are granulocytes. If you do not have granules, you are agranulocyte. A is without. So A. How do you know? Because there's not a granule in sight. Basophil, monophil, eosinophil, granulocytes. Monocyte, lymphocyte, not a granule in sight. So that's your mnemonic device to help you remember those. And these guys will have mostly nucleus, so barely any cytoplasm. You can see here there's barely, just a sliver of cytoplasm. Here, a little kidney bean shape, and then a little more cytoplasm. What is HIV and what is AIDS? What causes HIV? The virus. What causes AIDS? The same thing, but it's just defined as a white blood cell count below 200. So that's a T cell count below 200. So that means you have no defense. You have full-blown AIDS at that point. The virus done its job and it weakened you. Leukocytosis when it's high, leukopenia when it's low. Anything is penia is low.
So that means like HIV or AIDS is leukopenia?
Oh, it causes low blood cells.
Okay.
So in this case, low T cells. Platelets. Thrombocyte. So thrombo is clot, cyte is cell. So these are just fragments. So as the stem cells change, right, little bits kind of break off of the cells and they become your platelets. Most of the time they're not sticky. So they're circulating through your body in their non-sticky form. But when a clot needs to form, they get turned on and they become their sticky form. And you'll see fragments under the microscope as well. The liquid part. What's the difference between plasma and serum? Blood plasma, blood serum. You guys ever heard those two terms? Serum. So blood plasma is everything in it. Blood serum, we took out the clotting factors, so it won't clot. We can store it longer. So when you give somebody, usually the liquid part, you're going to give them the serum. All right, proteins, very important. Albumin is probably your most common blood protein. And if there's stuff in liquid, then the liquid stays there. So it helps keep fluid in your blood, helps keep your blood pressure up. So if you ever see a bag of like a yellowy thick stuff, like gooey infusing into somebody's vein in the hospital, that's a bag of albumin. Globulins, globular shaped proteins. For example, LDL and HDL. You guys ever heard of those guys?
Yep.
So the good fats and bad fats. So this is the bad guy. This is the good guy. Low density lipoproteins and high density lipoproteins. They circulate they circulate fat throughout your blood. Fibrogen. So this is the inactive form, fibrogen. When it becomes active, it's Fibrin, fibrin. Other things, amino acids, those are your building blocks of proteins. Sugars, anything that ends in OSC is a sugar. Glucose, dextrose, maltose. What are nucleotides? what do they make? Yeah, they're the building blocks of DNA. And you have different types of lipids. So lipids are different types of fats. For example, cholesterol, right? That's a type of fat. Phospholipids, glycerides, triglycerides, those are all the fats. Gases, most of the oxygen is carried on hemoglobin. Most of the carbon dioxide combines with the water in your blood to form carbonic acid. Have you guys ever experienced carbonic acid?
Isn't that when you come up from diet?
No, that's nitrogen. That's the bends. Carbonic acid, water, and carbon dioxide. So soda is carbon dioxide dissolved in water, right? So all the oxygen is carried by hemoglobin, but most of the carbon dioxide is dissolved in the water. So carbonic acid, does your blood pH go up or down?
Down.
It's an acid, so your blood pH will go down. Yeah, you guys get it now? So if you hold your breath, what happens to your blood pH? If you hold your breath, what goes up? CO2 goes up. That makes more carbonic acid, so your blood pH goes down. What happens if you hyperventilate? Then your blood becomes basic. Right? So hopefully that helps you with your discussion question and research. All right, what is an electrolyte? Let's see if you guys remember. How does your body make electricity? So give me an example of one.
Gas.
Potassium, sodium, those have plus charges. Chloride has a negative charge. Calcium has a 2 plus charge. That's an electrolyte too. Very, very important one. Non-protein nitrogen waste. So you have those waste products. Anything that's protein, your body doesn't store. So if you eat a lot of sugar, carbs, fat, your body stores it. But if you eat a bunch of protein, your body has to get rid of it. Your body has to break it down and you have to pee it out. So that's the nitrogen waste products. Good for losing weight, but only short term. Here's an image of a blood clot. Hemo is blood, stasis is to stop. So stasis, to make it stop. So here are the fibers that form, and you see some of the platelets and red blood cells that are trapped. So to form a blood clot, you need about 13 different steps, and there's 13 different blood clotting factors. If you're deficient in any of those, then that's hemophilia. You guys ever heard of hemophilia? Your blood can't clot, so any bruise, any cut, you'll bleed profusely for a long time. The treatment's easy. If we know which clotting factor that you're missing, we'll just give it to you. Right? Anytime you have a bleeding event, we can give it to you, and then hopefully you have a normal clotting. So what happens is, as long as the platelets and the thrombocytes are inside the blood vessels, they're okay. It's when they experience things that are normally on the outside, different proteins like collagen and elastin that's on the outside, that's when they get turned on. So the platelets become sticky and the clot cells become active and they turn on the platelets. So you get the stickiness, they build a clot, fibers form, and then you trap red blood cells and you get that clot. If it dries on the outside, you get a scab, and then the wound will heal. So, inactive form, because we don't want this stuff to making clots spontaneously. So we have to turn it to its active form. So it's activated once it's exposed to something outside of the blood vessel. Sticks to the damaged area because now they're turned on, and then it traps the blood cells and platelets. Embolus, an embolism. So it's any blockage. It could be caused by a particle or even air. So in the brain, they used to call it a CVA, cerebral vascular accident. Now it's just a stroke, right? Blood clot that travels to the brain. If it travels to the heart, myocardial infarction. If it travels to the lungs, pulmonary embolism. And if it's deep inside a vein, deep vein thrombosis. Right? So it's all the same thing, it just where it happens, they change the name, so we all know. The ABO blood type. So A blood type, the gene for A blood is dominant over O blood type, and the gene for B is also dominant over O. However, if you get an A and a B from each parent, the AB gene shows co-dominance. So AB would be expressed, whereas O is recessive, it won't be expressed. So what determines the actual phenotype? So what actually results from your genes are the blood groups, and it's determined by antigens on the surface of the blood cells itself and the antibodies that's in the plasma. So you have surface antigens here on the red blood cell itself, and then in the blood plasma you have the antibodies. Agglutination. Agglutination is the clumping of red blood cells, and that's due to the interaction of the antibodies. So the antibodies will match the surface antigens, and they'll all combine together and clump up blood. So that's why you'll need to know patient's blood type in order to infuse the appropriate blood. So type A. So on the surface of the red blood cell type A, you have A antigens. But in the blood, there's anti-B antibodies. In B blood, you have B antigens and anti-A antibody in the plasma. So if we take these two blood and mix them together, this antibody will detect B blood, and this antibody will detect A blood, causing agglutination. So you can't mix those blood types. In the blood type AB, you have both surface antigens, but you have no antibodies in the blood plasma. And then for O blood type, there's no surface antigens, but you have both antibodies. So you have the universal recipient and the universal donor. So O blood is the universal donor because there's no surface antigen for plasma antibodies to detect and causing agglutination. AB blood is the universal recipient. Since there's no plasma antibodies, there's no possibility of blood agglutination. So here's a summary of the blood types. So surface antigens. So this is what's on the surface of the red blood cells. A antigen, B antigen. Both A and B antigens. O has none. But here in the plasma, the liquid portion, you have anti-B for A type blood, anti-A for B. Here you have none, because if you did, you'd kill yourself. And then in O, you have anti-A and B. So it's very important that O is the universal donor and the only thing we donate are the RBCs. We don't donate the antibodies. We separate them. The RH factor. So the RH factor is an additional surface antigen. So it's on the red blood cell, and most people are positive for it. Some people are negative, and we'll talk about the problems that can come up if someone is Rh negative. So one example is with transfused blood. So if you are not Rh matched, it may cause a future problem. So let's say there is a patient who is Rh negative. They get blood that has the Rh factor. So this is an additional antigen where this patient will now build up an immunity anti-Rh. So the second time the patient gets a blood transfusion, that immune system has adapted. So this can occur with pregnancy as well. So that condition is called erythroblastosis fetalis. So as a fetus, red blood cells can cause a problem. So it's not the first pregnancy that's an issue. It's going to be the second pregnancy. So in this condition, the mother is Rh negative. The first child is Rh positive. So the mother detects the Rh factor, producing anti-Rh. So in the second pregnancy, the mother's body will now attack the fetus. So as long as we know about it, we can suppress the mother's immune system. So in A, we have the first pregnancy. Here we have Rh positive blood being detected by the mother. The mother builds up antibodies for Rh. So this is after the first exposure. And in the second pregnancy, the mother's immune system will now attack the child. As long as we know about it, we can prepare for it. Some common diseases and disorder. So anemia is without blood, so it's usually red blood cells. There's other types. Leukemia, so this affects the white blood cells. So it's usually a cancer because there's abnormal production. Sickle cell anemia we mentioned. So if you have both traits, your red blood cells will be enlarged in sickle shape. That can get stuck, especially at high altitude or low oxygen levels. However, if you are a carrier for the sickle cell trait, you're protected against malaria infection. Polycythemia. So here's an example where you have high amounts of red blood cell produced, whereas thalassemia is imperfect hemoglobin, so you have short-lived red blood cells. All right, that completes our discussion on the blood system. So we did the cardiovascular system, blood, and in conjunction with the cardiovascular system, we're going to go on to the respiratory system.